query was these, so I have these 4 genes, and then you can of course do pathway analysis with PathBIX on that. So so you can just paste the same genes into there, then see what you get. And as often happens when you don't have too many genes, there is very little overlap with known pathways. So you get, I mean, even though it doesn't require overlap, there's also not enough interactions with such a small thing. So then we can use FunCoup to extend the query set. So then I pick here the expansion depth 1 instead of 0. And do this, I think. Shouldn't take more than a second or two. So then, so I expanded a bit. I mean, so now I didn't expand it to 30 genes because then, well, you might get too many pathways. So I just picked the 10 nearest neighbors here. And then as Emma did, I did the same thing. I went to interactors and I pressed enrichment in PathBIX. And then I get this list of pathways. So from the extended gene set, extended by the network. And well, I mean, you can of course wonder, well, are there known pathways? Yes. So there are some known, for instance, the notch signaling. As 2 of them have that, and neurotrophin signaling. And indeed, that's what we get. The notch signaling is the top scoring one. And so, but note that there are, so these blue boxes here are the genes that actually also overlap with that pathway. So there's no less than 6 overlapping genes. But all these other pathways, they would not, so you can see the Q value of the overlap-based method is terrible. So nothing is significant really, because the overlap is just 1 or 2 genes. Okay, this one is borderline. So, and then you can, yeah, then you can find, so these extended things. So for instance, here is a cardiomyopathy pathway that turns out to be related to Wnt signaling, which is quite relevant for well, for notch signaling. Well, I mean, Wnt signaling is the one above here actually. And then you can see that there's quite a lot of connections then between the query and the green nodes and the pathway, the blue nodes. And the genes that are both the query and the pathway are sort of have both blue and green coloring. And yeah, you can go to the KEGG pathway if you want to learn more. But so these are all then reasonably significant things that you can find by extending the query in FunCoup.